Professor Duschl comes from Austria, from the University of Salzburg. And his presentation will covering delivery of nanoparticles, allergen conjugates to human immune cells, implication of allergen immunotherapy, and safety. Okay, all prepared?
All right. I am going to talk about allergy, and this is now real classical type 1 allergy, not the CARPA, which is a pseudo-allergy that we have been already heard about. Allergy is a serious health problem. In the developed countries, 20 to 25%. This is a standard, what people are saying. Actually, it can be much more in some areas. Record holder in Europe is Scotland. And it's not a genetic issue, it's a lifestyle issue where you have a high allergy. We have frequently co-exposure towards nanoparticles and allergens, a subject that has not been very much investigated. However, I'm not going to talk about this. We will deal here with medicine. And here the interest is in therapy of allergy, for allergen immunotherapy, and a number of groups have investigated this in the mouse. We are interested in human issues. When you are diagnosed with allergy, immunotherapy is the only causal therapy that is available. Everything else is symptomatic. So you get a high amount of your own specific allergen, and this leads to the generation of regulatory T cells and to the induction of competitive and protective antibody types. Essentially anything will work as long as it is not IgE. Of course, when you think about nanoparticles, one issue is that you can really load them with a very high amount of allergen, and another issue is that you can add additional ligands to the surface, like ligands for pattern recognition receptors and so on. In the human, we are looking at different allergens. Here in this study, we looked at the major allergens of birch pollen, of house dust mite, and of Timothy ryegrass. If you look at these allergens coated here to the surface of nanogold particles, it looks like a homogeneous surface, and indeed, if you look for the thickness of this layer, it's this one molecule thick. However, if you check the true amount of protein molecules bound to the nanoparticles and model the whole thing, you find it's not quite as homogeneous, it is not a full coverage, but still it's a lot of allergen molecules, and this certainly looks interesting if your task is to overload the immune system with a lot of the allergen. We can test whether these substances now are changed in the allergenicity. This is a safety issue for patients because you are giving a high amount of allergen. Naturally, this is dangerous, and there can be a problem with allergenicity. Maybe there is a change if you have it bound to nanoparticles. Now, one way of testing this is the basophil activation test. You use basophils from human patients. They come already loaded with a specific IgE bound to high affinity receptors. And if you now add allergens, you can look for upregulation of markers, and you can look for the release of mediators, which is what basophils would be doing in a state of anaphylaxis, for example. You get those strange curves which are familiar to allergologists, but nobody else. We have completely different results for the different allergens, so there is no general rule. For the birch pollen, we find some patients now show much higher allergenicity, others reduced. For some it doesn't matter. This can be explained because birch pollen allergen has epitopes all over the place. If you assume non-random bindings, this is expected. The house dust mite shows always a stronger response, And for the Timothy cross, there is no difference whether it's bound to nanoparticle or not. Naturally, this house dust mite is interesting. It's ubiquitous, it's perennial, and it's not just an allergen, it is also a protease. A surprising number of allergens are enzymes, and most frequently they are proteases. If you look for the protease activity of this protein, when coupled to nanogold, the response goes clearly up. It's now a much better protease, shown to the left by cleavage of a model peptide, and here by impaction on the tightness of a monolayer of A549 cells, which is a simple model for the alveolar epithelium. So where do you want to take this? Going into therapy, we are looking for particles that you can get in many different shapes and many different conditions. Particularly, we are interested in mesoporous particles. So we ended up with titania and silica. And when we couple the allergen, it's actually corona formation. Also in the study with the nanogold, we just incubate that stuff for 1 day, centrifuge, and in the pellet you find your coupled nanoparticles. I will get into it why we use this unexciting method. Actually, even the lowest amounts that we use give you a pretty decent coverage, and after that you only increase the allergen in the supernatant. So this is really simple. And if we look whether this material is taken up by cells, yes, it is taken up. The model protein here is GFP because you don't need to label that. You find the fluorescence inside of the cells. And when you are looking for 3D, yes, they are inside of the cells. They are not just on the surface. Now, since we are only doing a corona formation, there is no covalent coupling, there is nothing fancy going on, and the corona evolves over time. So we are looking at replacement effects where the allergen corona is getting substituted by other proteins at the latest when we add this to cell culture medium that contains FCS. So indeed, the amount of coupled proteins, whether it is GFP or birch pollen allergen or rhodamin-labeled birch pollen allergen, it drops even after 1 hour, a little bit more after 24 hours, so most stuff occurs early on. But still, we maintain about 35% of the target protein. We had a lot of the stuff on the particles, and 1/3 of a lot of stuff is still quite a lot. So this would still be a good amount. We have now to correct for the amount of proteins that remains bound. The amount of allergens that remains bound is not replaced by albumin or other components of the FCS. And if we do that, we find that uptake into cells is done with quite efficiently. After 1 hour, the significance is not yet statistical. I think it's true, but at 24 hours, we clearly have a higher uptake of the nanoparticle-bound allergens compared to free allergen. In case you are wondering, by the way, about this ugly nomenclature here, BET-V1.0102. This is now the official allergen nomenclature, and I hate it. It used to be BET-V1D and BET-V1A, and nobody can pronounce that stuff. And then in short talks, it's just impossible. Now, why are we doing that? I mean, corona formation is pretty old-fashioned, and it is low-tech, and it would be much more exciting if we use some fancy chemistry. We would be happy to do it. But what do you get when you are undergoing allergen immunotherapy? That concept was introduced in the 1920s, and it was originally done with allergen extracts. So you get cat hair extract. For bees, they got dead bees actually, as the allergen source. Plant pollen, you have to collect for birch tree, you have to collect it from a tree. And don't ask me where they get house dust mite. It's actually house dust mite farms, and they collect the excrement and extract that. So you can be a mite shit farmer. There's a job actually exists. And this is still what is used. Those are still extracts that people are getting. And of course, now there is recombinant proteins, but they are not in the clinic. First of all, the extracts are cheaper. They're in the clinic for a long time, and they are natural. So you don't have to spend a lot of money to get them approved. You use a recombinant protein, it's treated as a drug, and you have to go through extremely expensive processes. There are clinical trials, but nothing is in the clinic. If you could use extracts that are already in the clinic, I think it is much more reasonable to expect that you can make it if you combine it with nanoparticles. And indeed, this works. Here's extract from birch pollen, the major allergen, as a monomer, as a dimer. Coupled to silica, yes, it is still there. Coupled to Mesoporous silica, yes, it is still there. We are betting a bit on the Mesoporous for delivery and a bit of a depot effect. Rhodamin labeled, no, that did not work. But this is the direction where we want to go. I would like to conclude by stating that the allergens bound to nanoparticles can have a changed allergenicity, which has to be taken into account when considering safety of such treatments. Enzyme activities can be affected, and indeed, if you have an enzymatically active allergen, you have to look for this. The basophil activation test is suitable here, and in our opinion, the simple corona formation is an interesting concept because you can use those quite dirty allergen extracts that are already in the clinic, which makes it more likely that a nanoparticle formulation can make it too. I do thank our funding agencies, I do thank my group members, our cooperation partners, and of course I thank you for your attention. Thank you.
Thank you very much, Professor Duschl, sharing your data with us. Any question to our speaker?
Yeah, I have a question of it. So is the idea then that you want to get these allergen proteins delivered per se in order to induce an immune response, because the direction of the immune response is what clearly is going to determine the outcome. In the world of practicing allergists, we practice this voodoo art with extracts in the hope that we will induce immune tolerance as an outcome. Simply delivering the antigen on a particle surface is not going to be instructive as to the type of immune response that's going to follow. So is the assumption that if you put it on the mesoporous silica and it gets taken up in whatever the vaccination site, is that it will do the job of instructing regulatory T cells? Is that the aim? Or do you want to induce an alternative immune response where, say, you know, the IgG then becomes a potential blocking antibody?
The delivery here is to antigen-presenting cells, mainly dendritic cells, and you can target the allergens in a more controlled and probably more efficient fashion to disease if you pick the right size and other properties. So the delivery into the target cell may be better, and the loading of allergen is well controlled, and you get high amounts of allergen into the antigen-presenting cell. So that's the idea, that the dendritic cell then instructs T cells what to do, and if you load with a very high amount, this is supposed to result in protective responses. You can add to the nanoparticle additional agents like pro-inflammatory agents, which are anti-allergic. However, this would be an additional modification, easily done, but then you are not longer with these classical extracts that are now clinically used. But we need to get into the dendritic cell.
Yeah, but how do you ensure after you got into the dendritic cell that the direction of the immune response will be appropriate? Because if you take the protease active antigen on a particle surface, there's also the innate lymphoid system that we learn about now, where those allergenic enzymatic activities may trigger at that level already an immune response that could take you to a place where you don't want to be, and that is not primarily directed by a dendritic cell.
It will be a problem if you look at something like DERP1, where you indeed have the protease problem. It's not an issue for birch pollen, where you don't have proteases. And I do not assume that it will work perfectly, but allergen immunotherapy is a bit of voodoo and does not work that well as it is. If we get a small improvement, it would already be good for a lot of patients.
Yes, you're just doing a little marathon, okay?
Joining question, do you follow IgG or IgM following your treatment?
We are doing this with human cells, and in the human cells, of course, you don't look for antibody production. For that, you have to go in the mouse. But I have a feeling that we need to understand the human cells' behavior better, so we don't follow it up to the level where we have antibodies. What we do is co-culture of dendritic cells and T cells, so we can look for the cytokine profile. And when we see the cytokine profile and the transcription factors that come up in the T cells, we have already a pretty clear idea which antibodies will come up. However, T cell response was not determined in these special studies that I have talked about.
Anybody else?
Have you done some experiment, some studies to understand how the protease interact with the surface? I'm asking you this because it seems that almost at some extent, some of these proteins remain irreversibly absorbed. So it means that surely some conformational modification occurs. So probably the object at the surface is different from the native one.
Yeah, we do assume non-random binding, and we do assume change in structure of the protease. However, we have no data on that. The project is going on to study exactly that specific question. Of course, this is of tremendous interest for us.
Okay, thank you. So now we're looking forward to the last speaker of this morning.